Good morning, everyone. We're going to go ahead and start it. It's my great honor and privilege to welcome Dr. Loren Wold. He's currently a professor in Ohio State University with joint appointment in several departments and colleges. And he's a director of biomedical research at College of Nursing, and also vice chair for faculty affairs in Department of Physiology and Cell Biology in the College of Medicine. And he grew up in our great neighboring state, North Dakota, and went to Boston University for his undergraduate study, and then came back to North Dakota, taught about a year in University of Marist, and then decided to pursue his training in science. Got his Master of Science degree in 2000 and a PhD in 2003 in Department of Pharmacology, Physiology, and Therapeutics at University of North Dakota. And then after his postdoc training in University of Southern California and the University of New England, he took a faculty position in Ohio State University in 2006 and rose to the rank of the professor in 2014. I think Loren is very active in several professional societies and served in multiple grant review sessions, including NIH study section, and won many honors and awards. His research is well funded by NIH and also American Heart Association. His research is focused on how the environment affects the health and the function of the brain and the lung and the heart. We share a common interest to study how the environment factor affected cardiac maturation during the neonatal development. And he has like 85 peer-reviewed publications. Today he's going to tell us what we breathe in can affect our overall health. Welcome.
Great. Thanks so much, Dr. Thank you very much. Great memory. I was impressed with that. So it's great to be in Minneapolis. Growing up in Bismarck, this was the big city, and we used to come here. My grandma lived just about 2 miles from here. So it's fun to be back, and it's an honor to be here. So I know we have to be out of here by 9, so I'm going to get going. But I wanted to tell you a little bit about some of our past work and recent work, some unpublished today, and really walk you through what we've been working on in the lab for the last couple of years. So we know air pollution's bad. This really became pronounced back in the 50s during the London smog, when around 18,000 premature deaths were attributed to exposure to particulates in the air. We now know today, of course, that it's still bad. Oops. Is there a pointer? Yep, right here. So this is Beijing, but what's unfortunate is this is Los Angeles. So as you can see, this is not just a problem for other countries. The United States has just as bad of a problem, and I used to think that growing up in North Dakota we were immune from, of course, any effects, but the problem is during farming season, all the stuff that's put on the crops gets re-aerosolized. So what does air pollution consist of? So we know that air pollution consists of many different things: gases, liquids in suspension, and particulate matter. So we're going to focus today on particulate matter. And what is that? Particulate matter contains a carbonaceous core surrounded by heavy metals, organic molecules, nitrates, sulfates, etc. There's 3 types of particles that we're going to talk about. The big particles are the PM10 or coarse particles. Those particles traditionally get caught in the upper bronchi and don't penetrate too far into the lungs. PM2.5 or fine particulates around the size of a red blood cell, these are produced by predominantly by the combustion of fossil fuels. And we also know that these are the predominant particles that affect human health. And then the ultrafine particles, or 0.1, which are around the size of a virus. The problem with the ultrafine particles is they actually can group together, they're sticky, and they group together and they can produce or become a fine particulate. So in 2010, Circulation published an article that was a combined statement from the American Heart, American Diabetes, and several other organizations that said PM affects human health. And what you can see from this graph is that anything above the hatched line showed a positive association between exposure to particulates and all-cause mortality, cardiopulmonary disease, CVD, and ischemic heart disease. So PM exposure has been positively associated with MI, atherosclerosis, arrhythmias, thrombosis, alterations in blood pressure, and an area we've worked on, which is hypertrophy. So some of the questions that we've focused on in my lab is, how does exposure to PM affect the heart? So we can imagine, of course, it's going to affect the lungs. That stands to reason. But how does this potentially have an effect on the heart? Does exposure to PM work synergistically with other factors such as obesity, hypertension, etc., that also can cause cardiovascular disease? And does exposure when we're young affect us when we get old? So some of the mechanisms that we've looked at, both indirect and direct. So the indirect mechanisms are those that the lungs are affected, resulting in inflammation and oxidative stress. Stuff gets into the bloodstream, then affecting the heart. But what about those particulates that actually can penetrate into the bloodstream? And so it's been shown in humans who volunteer and get a $50 Target gift card to stand in a box and breathe what is called inert air. And what they do then is just take blood samples, and they can see that these gold-labeled particles are in the blood. So we know that they can actually traverse the endothelial lining and get into the bloodstream. So these are the main mechanisms that my lab has really looked at. So there's multiple models that are used to actually administer particulates, and so some labs will actually put a tube down the animal's throat in order to instill it, or use nasal inhalation. We use an inhalation method where the animals are running around happy, breathing and eating, and don't know that they're breathing air that's dirty, if you will. So in the studies, and I'll show you a picture in a second, but the studies where we expose mice to relevant concentrations of PM, what we do is we actually expose the animals to concentrations of particulates that people were exposed to in Los Angeles the day before. So we use sort of a paired exposure regime. We expose them for 6 hours a day, and the equivalent 9-month exposure sort of safe concentration is 15 micrograms per cubic meter. So we're exposing to concentrations that are considered safe by the National Ambient Air Quality Standards. So this is actually our exposure setup. It's all man-made. Unfortunately, they don't sell these. So what we do is we bring in air right up here from outside the Columbus region. It's concentrated, filtered, and exposed to animals in these two compartments, which you'll see here. So you see that the animals can move around. They are not restrained, so the stress levels are much less than if you're anesthetizing them, etc. So this is to show you over almost a year course the alterations in particulates. And so the area where we actually expose the animals is near the intersection of 3 large freeways, interstates, within the city. And what you'll see here is there was actually a building that was being built near the site. So Ohio State, we're a big place, and we rent buildings, and this building was in an industrial park. And so this, we call this the industrial effect or the university effect, because there's always buildings going up and going down. But you can see that it doesn't remain stagnant. It's up and down over the course of multiple weeks, and what we do is we just present an average. So this is really where it all began many years ago. I like to tell the story, especially for students. So I got a call from a colleague at Ohio State main campus. I, at the time, was at the Children's Hospital, which was about 10 minutes away. And a colleague called and said, hey, we've got these mice. They've been exposed to particulates. We're done using them. They had done some behavioral tests. And now we don't have any more use for them. Are you interested? And so I had no clue what we were going to do with them, but I said, of course, because they're free. So we took them. We did just basic echocardiography, and what we showed that 9-month exposure to concentrated particles, simply control animals, or the filtered air component, which is a group where filter is placed at the inlet to filter out all particulates, we showed a significant reduction in fractional shortening. So just exposure to those particles long term caused a significant effect functionally in the animals. We saw no change in heart rate, and you might be thinking, what about blood pressure? We did see a significant change in mean arterial pressure, but it was only on the degree of about 4 millimeters of mercury. So we didn't think that that could explain the significant change in function. My lab, so I'm traditionally trained as a cardiomyocyte physiologist, so I'm interested in how the single cells of the heart can function in real time, and if that can recapitulate the functional or the in vivo setting. This just shows a representative twitch or contraction of the cells. And what you can see here in this graph is peak shortening. This is the cellular equivalent of fractional shortening or function in the whole heart. At control levels, these are myocytes taken out of those animals exposed for 9 months. We saw a significant reduction in cell function. Interestingly, if we can do a stress test in a dish, you might be familiar with dobutamine stress tests given to humans, but what we can do is actually put isoproterenol in a dish, which is a beta-adrenergic agonist, and we show that there was a hyper-responsiveness. And so we'll get into that as we go along. Time to relengthening is the time that it takes the cell to get back to normal, and time to contraction listed over here. So we saw that the in vitro component recapitulated what we saw in the whole animals. We also look at calcium. At control, there was no difference in basal calcium levels. So if you don't know, so calcium is very involved in contraction of the heart. But interestingly, with ISO, we saw, once again, a hyper-responsiveness. So once again, this is really where it all began. We looked at a bunch of different proteins involved in calcium signaling, and I'll focus your attention here where we saw a significant increase in the phospholamina to SERCA ratio, which basically showed that calcium regulation within the cell was altered. We collected serum from these animals, so we know that inflammation is involved, but we collected the serum 18 hours after the last exposure and looked at 38 different inflammatory markers, and I'm only going to show you a couple here. And the reason is because every study we've done, and we've done lots of studies where you just take cells and expose them to particles in the buffer, etc., we see that MCP1, or myocyte chemoattractant protein 1 and 3, are always up. So thankfully, and the CCR is the receptor. So we're finally now exposing knockout animals to MCP1. So just this first part, conclude that long-term exposure to particles is bad. It affects the heart. The cardiomyocytes taken from those animals have reduced contractility associated with altered beta-adrenergic signaling. And we saw an increase in inflammatory markers. So that's a long-term exposure, and in mice, 9-month exposure is a significant portion of their lifespan. We also exposed obese animals. So our hypothesis was, as I stated before, earlier, that obesity, as we know, can also cause heart disease. So we were interested in, does particulate matter exacerbate that? And if you follow this line of fractional shortening or contraction of the whole heart, you can see that at 3 months there was no difference, but by 5 months there was a significant change in the hearts from obese animals. And so if any of you have seen, these are the Obese mouse, which is a defect in the leptin receptor, and these animals get massive. And so it stood to reason that there was a significant reduction by 5 months, much earlier than what we saw in the controls. We also took myocytes from these animals and showed that, once again, the in vitro component recapitulated what we saw in the whole animals. And so this stood to reason with our hypothesis that myocytes were altered from these animals, and we also saw some changes in the times to contraction. So that's great, but that's really in adults. What about exposure in utero? So we know that in epidemiological studies, PM exposure has been shown to increase the prevalence of low birth weight, infant mortality, and preterm birth. We also know in some clinical studies, particularly in Asia, that humans that live in very polluted areas for the duration of pregnancy have the offspring have multiple changes, which I won't get into. But in one study, mothers in areas with high pollution had infants weighing 35 grams or less on average than those who did not. So animal studies also recapitulated what we saw in humans. So diesel exhaust particulates exposed prior to pregnancy decreased birth weight, increased abortion rates, and caused altered development of female offspring. This was coupled with changes in the placenta, as well as altered oxidative stress, in this case, myocardial lipid peroxidation. So we knew that the animal studies were recapitulating what we were seeing in the humans. So this, of course, is looking at the fetal origins of adult disease, or the Barker hypothesis, where our question was, how does PM exposure affect the cardiovascular system during development? And so in this initial study, we really wanted to look at capturing a wide range of development. And so we bred the animals in the compartments, which you saw before. Once they gave birth, we began the exposure, and then the animals were exposed all the way until weaning. Okay, so quite a large exposure window, but once again, we just wanted to capture that major development, all the developmental stages. The pups were removed from exposure at weaning and left alone until 3 months or early adulthood. So we really wanted to just focus on exposure during development. And then we looked at echocardiography, once again, cardiomyocyte functional changes in pressure volume loops, or ex vivo, where we implant a catheter into the actual LV and look at pressure development. So this is the data from the study. At birth, we didn't see a difference in the number of pups per litter or male-female ratio. What we did see, there was a significant reduction of birth weight on average of the pups. But at adulthood, there was a significant increase in body weight. And so we're seeing this now in some clinical studies done in Asia, where children who live near very polluted areas in industrial settings are at a greater risk of metabolic syndrome and insulin resistance. And so we're seeing obesity increase in those areas. We also saw the heart rate to tibial length, or our measure of hypertrophy, was significantly increased in these animals as well. So the heart data from this, functional fractional shortening here, just M-mode echo, showed a significant reduction in these animals at adulthood. So at weaning, remember, we stopped the exposure, and then we looked at them at 3 months of age, and we saw that there was a significant functional change, which was coupled with some changes in structure. So over here you see the LV end diastolic and end systolic measures, which were changed. We also saw a significant reduction in posterior wall thickness. So pressure volume loops, once again, we put that catheter into the LV and looked at pressure development. So ejection fraction was significantly reduced, along with what we saw in fractional shortening. We also saw significant changes in dP/dt max, and this was coupled with increased end systolic volume and end diastolic volume. So the hearts, when taken out of the in vivo setting, were also altered. The myocytes from these animals were significantly changed, just very similar to our other long-term phenotype. But what was interesting is we only saw a significant but minor change in the time to contraction, which was not evident in the time to relaxation. So these cells were affected, but it seemed, since calcium is what's regulating the contraction and relaxation, that calcium was not as altered as in other models. In this figure, what we're showing is actually isoproterenol. So when given iso, we normalized the peak shortening. And so what this is telling us, once again, is that the calcium signaling pathway is altered but is fixed, it seems, with beta-adrenergic stimulation. Calcium was not different at baseline, but tau, so tau is a measure of fluorescence decay rate, and so we look at calcium through fluorescence using FIRA as a dye, and you can see here that the tau was increased, so the rate of decay was significantly increased. Another question we had is fibrosis involved, and so using just Picroceria spread staining, we saw that there was a significant increase in PSR in just heart slices from these animals.
Does that persist as the animals get a little older?
Yes, so we've looked, this once again was just at 3 months. We've looked at them at 6 and 9 months, and it gets much worse.
Really?
Yeah, so these animals are sort of doomed. And I'm going to show you a bunch of different other windows too, and we see that consistently, that it just seems to get worse. Even if you take them out of that and put them in filtered air, it doesn't revert. Yeah, good question. So in this study, perinatal is what we call that exposure window. Exposure to normal levels caused reduced birth weight. Mice exposed during development had reduced cardiac function and increased collagen deposition, which was caused in part by reduced cardiomyocyte contractility and calcium handling. But once again, that was a large window that we looked at of development. And so what we wanted to do now is start teasing out that those developmental stages. And so in this work, we exposed just in utero. So we started exposure once we found the plug. If any of you have not worked with mice, we breed them, and once we find the plug, we know that 90% chance that pregnancy has occurred. And so it's a 3-week gestation period. We stop exposures once the pups are born. They still are nursing at that time, and we allow them to grow all the way up to adulthood at 3 months, and then continue the same measures that we've seen previously. And so this slide is just to compare. These are the groups I just showed you, the perinatal, where we saw that decrease. But if you stop exposure when the pups are born and don't touch them again till adulthood, we saw a very similar phenotype, where there was a significant reduction in fractional shortening. So this is just to show you the M-mode echo, representative echoes on the top, but you can see here that this is the fractional shortening that is the same as I showed you right here. We also saw significant changes in the structure, so the left ventricular end systolic and diastolic measures, as well as decrease in posterior wall thickness. We looked at pressure volume loops again. You can see over here contractility graph. The solid bars are the pups that were exposed in utero to particulate, and the open hatched on top are those from the controls. And you can see that contractility was significantly reduced. We took myocytes once again from these animals, showed once again that there was a significant reduction in peak shortening. And so the cells were recapitulating what we were seeing from the in vivo animals. We did not see any changes in time, as you can see here. So it seems that in developmental exposure does not have the effects, does not have as severe effects on calcium regulation as it does with long term. We were also interested in looking at the electrophysiology of these cells, and so we looked at action potential duration, how long the action potential lingers, if you will, and you can see that as we increase the stimulation of the cells, so we stimulate these starting at 3 hertz up to 5 hertz, or more physiological heart rates, you can see that there was a significant increase in the duration of the action potential in cells taken from those animals that had been exposed in utero. We also looked at multiple inflammatory markers. We saw that the interleukins were up, TGF-beta. We also saw increases in collagen-1 messenger RNA, as well as MMPs. And so we're seeing that persistent fibrotic phenotype as well. We looked at multiple contractile proteins. Circa is a major contributor to calcium regulation, and you can see here that circa protein was significantly reduced, as well as phospholambin, which helps to regulate calcium entry and sodium calcium exchangers. So these are all involved in calcium regulation of the cells. So that's great. We know that just exposing in that in utero period is bad, but what about their ability to exercise? Was there any change in exercise ability? We did this study because we had just recruited a new person to our department who's an exercise specialist, and so we're trying to establish a new collaboration and got some really cool results where we actually put these wheels in the cage and just allowed the animals to exercise as they wished. What we saw was even at just 1 week, there was a significant reduction in the kilometers traveled in the animals that were exposed in utero to PM, or the closed bars. Same was evident up to 3 weeks. And you can see here the average over 3 weeks was 17 kilometers in the PM in utero exposed versus 40. So it was a major reduction. We don't know if that was simply because they didn't want to, right? They could be depressed. Who knows? Right. They could be a Packers fan. I mean, who knows, right? So anyway, we haven't looked at that, but all we know is that they exercise much less. We also looked at echocardiography from these animals at the different weekly time points, and saw that they're consistently, except for it seemed at week 2, there was a significant reduction in contraction of the whole heart of these PM exposed. So that's great, but what about if we even take this further down? So before we were in the in utero, we were looking at that 3-week gestation. What if we expose just during the first trimester, which, as we know, is when most of the heart is developing, right? So we expose simply for 7 days. So this group is where exposure was stopped simply after the first trimester. They were never exposed again. The pups were born. We allowed them to grow to 3 months of age and did very similar studies. Lo and behold, we saw the same phenotype, that just during those 7 days. I'm not going to show you the 2 and the 3 and the 2 and 3 combined. What I'll tell you is we don't see the significant effects at second trimester alone or third trimester alone. It's just during the first trimester. We saw the same things with structure as well as decrease in posterior wall thickness. So just during those 7 days, and once again to remind you, we're exposing to levels that are considered safe by the National Ambient Standards. So this was quite, that's the same slide of, I don't know why I have the same slide, but this was quite striking to us. And we saw the same effects that I'm not going to show you with inflammatory markers, etc. So that was pretty crazy. So just 7 days exposure. We've done some studies where we've done just 1 day exposure. I'm not going to show you all that because they're incomplete studies. But what I want to do now is to talk to you about a recent paper of ours that just was published a little over a month ago. Actually, was just interviewed by NBC News 2 days ago. They came out to interview. It's going to be on in May, so if you see me. We were interested in, what if you expose animals before mom and dad meet, and then look at the offspring, okay? So step back a bit. I collaborate with one of my best friends who works at the VA in New Jersey, and so he sees a group of soldiers before they go to war in Iraq or Afghanistan and after they come back, and he looks at their cardiopulmonary health. And he was seeing some dramatic changes in their mostly in their lung function. He's more focused on the lung. So we were starting to talk about how could we do this in an animal model. You may have heard of the burn pit exposures that have gotten into the news a lot lately. So we know that the soldiers are exposed to very high concentrations of particulates because what they use is burned very close to them. So what we did in this paradigm is we exposed mom and dad for 3 months in separate settings. We stopped exposure. We introduced mom and dad. We very quickly had litters, and then we raised those pups in room air. It was in filtered air. They were never exposed to particulates for 12 weeks, so up to our 3 months or young adulthood. And then we did all of our assessments. What we were interested in was really the potential epigenetic changes that were associated with this exposure. So birth weight was not different. You can see here. Body weight at adulthood was significantly decreased. And I will say that this is all males, and I'll tell you why that is at the end. So all the offspring data I'm going to tell you is in males. Body surface area was decreased. Heart weight was decreased. And heart weight to tibia length was also decreased. So these pups who were never themselves exposed had changes. They were hypertrophic. What's interesting here is look at the fractional shortening. So these hearts from these offspring, male offspring, had significant dysfunction at 3 months, coupled with ejection fraction changes. We also saw, we did not see the changes in the LVES or ED, but we saw a significant reduction in posterior wall thickness. So structurally, they were also altered. We looked at the cells from these guys. And lo and behold, the cells also showed significant reduction in function. So we had an in vitro dysfunction that was evident from these hearts, with no change in calcium levels. However, tau was increased, so the fluorescence decay rate of calcium was increased, which meant that it took longer for the calcium to go back into the sarcoplasmic reticulum. We looked at multiple different proteins. We saw that there was a significant increase in circuit 2, coupled with phosphorylated phospholamban, with no change in sodium-calcium exchangers. So calcium was also altered in this setting. We saw, we looked at different oxidative stress pathways. We saw changes in nitric oxide. NERV2 was increased. Superoxide dismutase was increased with no change, no significant change in catalase. So oxidative stress was also evident in this model. We looked at the inflammatory markers. We saw the interleukins were up, TNF-alpha was up, CRP was up, NF-kappa B, etc. And here, our little favorite guy, MCP1, was also up, and I'm showing you the representative blot on the side here. But MCP1, once again, is something that comes up in every model we look at. We then looked at a global epigenetic marker, or DNMT1 and 3A, so DNA methyltransferase, and we saw that there was a significant reduction in DNMT1, coupled with increases in the sirtuins 1 and 2, which are really a global. We wanted to just do a global assessment in the heart, so heart homogeneous, to see if epigenetics was at play. So what I will tell you is we are now doing the male-only exposure, so dad-only exposure, mom-only exposure. The backstory for wanting to do this was there was an RFA that came out. We had 5.5 months to get some data, so we wanted to do a mom and dad both exposure just to see if we would see anything at all in the offspring. So we're cycling back now and doing the dad exposure and also looking at the sperm as well as the eggs to see what epigenetics are at play with germ cells, so we can try to link that to cardiovascular abnormalities in the offspring. So what I've done here is just plotted all the different models that I've shown you to show you the similarities. So perinatal exposure, or that really large window, caused a significant decrease in function. The in utero exposure, first trimester, and preconception. Quite striking that they were all pretty similar in their outcomes. So overall, PM is capable of causing contractile dysfunction by both direct and lung-mediated or indirect effects. Long-term exposure is bad, as we showed you in the beginning. Exposure to particulates during development causes reduced cardiac function in adulthood. And the most striking, at least that I can see, is that exposure to particles can actually predispose individuals to developing cardiac dysfunction. So the people that actually do all the work, my current lab listed here, most of this work was, about 70% was done by an outstanding postdoc, Vanita, in the lab. Collaborators, actually collaborating with a former professor, which is kind of fun. Studies I haven't shown you, which is a large portion of our lab right now, is actually looking at the interface between Alzheimer's disease and hypertrophy and cardiac hypertrophy. So what we had shown is in these animals, so we of course take every organ there is. In the long-term exposed animals, the brains, we actually see aggregation of APP, or amyloid precursor protein, which is the mouse analog of beta amyloid. We had also done behavior tests, and these animals, when exposed to concentrated particulates, have a behavior phenotype that's very similar to the APP knockout mice that are the Alzheimer's, if you will, mouse. So we have a grant from the National Institute of Aging right now to look at the hearts of Alzheimer's animal models, working in collaboration with Federica Del Monte, who just moved from Harvard to MUSC in South Carolina, where she's shown she showed in JACC last December that patients with Alzheimer's on autopsy, their hearts have aggregations of beta amyloid. So that's sort of another ancillary study that we're working on. We're also doing all these similar studies with e-cigarette exposure. So particulates, of course, are in these e-cigarettes that are all the rage now. And so we're looking at the effects of that as well as in utero exposure, et cetera. So just showing you some of the particulate data, and of course thanking our current funding sources here, and oldie but goodie picture of the lab. So with that, I will take any questions you have.
Fascinating talk, raised a lot of questions on my mind. I'll start with two, if I may.
Yeah.
You said that you have been able to demonstrate these PMs in the circulation. Where do those PMs end up? Do they end up in the liver, or do they end up in another organ? And the second question is, for those of us who might be doing autopsies on patients who have heart disease where we suspect that this might be due to PM exposure, what do we look for in pathology?
Yeah, that's a great question. So I will say that the studies where particulates were found in the blood were done in humans by a collaborator of ours in Washington. That was many years ago. We've done some tagging of particles, so you have to actually gold label the particles to tag them, and using electron microscopy have found that for some reason the particulates aggregated near the mitochondria within the cell in the heart. We have not looked at all the other organ systems. Others have shown that they're localized in the brain as well as the liver. So, but as far as it would be hard to find the particulates if they're not labeled, is the problem. But I can imagine that, I'm just trying to think of what exposure paradigm would already be labeled. You'd have to look for some, it's a good question. I have to think about that, because you'd almost have to label them to find them.
And what do we see in adult humans who might have this disease pathologically?
So, I mean, you're most likely going to see significant cardiac issues, hypertrophy in particular.
And fibrosis?
And fibrosis, yeah. Which is, it's going to be hard because you know, you're going to have high blood pressure induced changes, et cetera. So, yeah.
So we're all paying the price for where our parents lived and the sins they committed. while there. Have you gone to the grandchildren?
Yeah.
And
Yeah.
because you've got an epigenetic stable change that
Yeah.
obviously was in germ cells, and how far does it go?
Yeah. So we haven't looked at the F2s yet. We had just actually finished this study and put the grant in, and it got trashed because they didn't believe any of the data. They said there's no way that you can pass this along, and so we've done some epigenetics now and looked at sperm motility, etc. We're planning to do the F2s with the male only. We anticipate it's most. So I'll back up. So there's some literature showing that high-fat diet, that dads who consume a high-fat diet can pass along a predisposition to cardiac issues in adults, offspring. We're anticipating that this is predominantly a male-driven epigenetic phenomena. So we're planning on doing the F2s from that. If we don't see an effect with the offspring of male only, then we'll have to go back and do the double. So, but yeah, it's, how many generations could this potentially be passed? Who knows? So it's pretty scary. Yeah, we were shocked at how robust of a change we saw.
Yeah, pretty frightening. But
Yeah.
Yeah,
and these
certain countries in the world where there's like the most incredible air pollution
Right.
you've ever seen.
Right. And I will say these are all blinded too. So our exposures, it's not like the air you can tell is dirty. So these are all blinded studies. So it's pretty shocking. The reason I said we only looked at male offspring, females are protected.
Really?
Female offspring don't have any cardiac issues. However, if you stress them, we've done MIs and TAC, they are significant, so all the animals die. So, good question.
So have you looked at the inflammatory marker in the blood? Is anything changed in the blood that you can monitor and compare with the change in the heart?
Yeah, so the long-term stuff I showed was blood inflammatory markers, all the interleukins and TNF. We use this, mesoscale, ELISA, 38 different inflammatory markers. And then we also looked by PCR at inflammatory markers in the heart tissue, and we see very similar effects. So, as far as I think you're getting at sort of a therapeutic type.
Yeah, I'm just
question?
wondering to see if any serum markers, especially they look like the aging has alpha is looking for environmental effect and telomeres, and then how affected the cardiovascular health and maybe aging too. And
Yeah.
how you look at like a blood, like a telomere and other things.
Yeah, we haven't done any of that. We do consider this a sort of a model of early aging,
Yeah.
if you will, because all the endpoints are very similar to aged animals. So, but we haven't looked at telomeres at all. So.
Yeah, fascinating talk. There's a lot of questions. You may have shown it, but there was a lot of data.
Yeah.
Blood pressure?
Yeah. So you sound like one of our reviewers.
I didn't review it.
So the problem is we do tail cuff, which basically in a mouse you're just you're putting a little constrictor on their tail to measure blood pressure. It's not the most accurate. It gives you a sort of a window. The best way, of course, is to do implantables into the animals and to monitor them over time. We have too many animals going at once that we just can't afford it. I will say the long-term exposure did not cause a change in systolic pressure. It caused a change in diastolic, which then caused the mean arterial to be significantly different. It was up. But it was only like 4 millimeters of mercury. So we didn't think that could actually explain the hypertrophy that we were seeing. But others have shown that blood pressure is involved with higher concentrations of particulates. So the average concentration in Columbus is around 8 micrograms, which is pretty low. But we do it, so we actually concentrate 10 times. So we're exposing to 80, if you will. It can get up to 15, so then our exposure is around 150. So, you know, if you're in Beijing on a normal day, it's 100, right? So it's, so blood pressure is involved, yes, but I don't know to what extent.
What, I should know this, we have he's probably not here, Steve Heck is kind of a world's authority on smoking and changes that occur in the DNA and whatnot. You know, the thing that's curious to me is what would a cigarette or a pack of cigarettes as far as particulate exposure be for you?
Yeah. So the particle people hate the smoking people, and the smoking people hate the particle people. I will say that I'm of the opinion that a particle is a particle. I'm more interested in just itself as a sized particulate, right? So I will say that most of the data is quite similar to smoking data. The concentrations of particulates that a smoker would be, you know, so the first pass, if you will, or the smoker themselves are going to be exposed to is exorbitantly high, but usually from smaller time points. And I'm saying that just because I don't exactly know the particle levels that are exposed. I will say that we're doing these e-cigarette exposures now. And we're exposing to, so we basically make if you've heard of these jewels that are all the rage. My sister is a 4th grade teacher in North Dakota, and she finds them, her students, 4th grade, have these. But we're using the constituents that Jewel uses and exposing animals. And the particulate levels get upwards of 250 micrograms of particulates. But I'll give you the foreshadowing of the data. We 3 months exposure to animals, we see no effect on the heart. Hope no one from Jules in here.
Can I go back to the experimental design? You said the air these animals were breathing was from an intersection somewhere in Columbus.
Yeah.
How do you tease out particulate from other gases that are in the air?
So we don't. So we look at it as just a heterogeneous mixture of gases, particulates, dust, etc. We're not looking at the individual components. There's some groups in Louisiana with the Superfund that are looking at individuals. But my argument would be that you're never going to be exposed to just one thing in a real-world setting. And so I'm dancing around your question because we don't separate them out.
So it's possible that some of these effects are due to nitric oxide and other...
Sure.
Nitrous oxide.
Sure, exactly. Yeah.
Just curious about the females. You say that when you scrap, the female has a phenotype. So you think that the females, because they are compensated better, or maybe they're born with less male title somehow, then...
We've stayed away from the females.
Okay.
They're too confusing.
Yeah.
No, so we actually just got a supplement funded recently to look at the females in these because we're looking at estrogen as a protectant.
Yeah.
Yeah, but it confused the matters.
Any more questions?
Just a comment on North Dakota. He's not in the room. He's retired, emeritus professor Vince Gary.
Yeah.
He actually way back was showing chromosomal abnormalities in veterans who were exposed to Agent Orange. Actually, the DNA suppressed it.
Yeah.
But the other interesting thing he did was he looked at fungicide and herbicide applicators in North Dakota, South Dakota, and low birth weight kids, increase spontaneous abortions, and then also lymphomas.
Yeah.
Somebody at NIH found a gene that gets damaged and whatever, so it's a present danger outside the city.
Right.
Obviously.
Well, so I remembered back when I was in graduate school, there was a woman who was in the anatomy department, and her master's thesis was looking at kids who rates of autism or ADHD back then, and how close their houses were to the fields on the farm. and there was a striking correlation between those. So now you fast forward, and we're seeing brain changes, that are very similar to Alzheimer's. There really isn't good autism measures in animals, but I think it's all sort of related. So it's pretty crazy. Well, thanks for coming out so early.
Thank you.